The internal transcribed spacer 2 ribosomal gene, or ITS2, is one of the most important markers in molecular systematics. Research of the last 20 years has mainly focused on exploiting the ITS2 sequences for phylogenetic reconstruction. However, the secondary structure of ITS2 is increasingly being used in the field of phylogenetic inference due to several reasons. Secondary structure analysis extends the taxonomic applicability of this marker. Whereas the rapidly evolving and therefore very variable ITS2 sequence is mainly suited for phylogenetic analysis at species level or lower, adding structural information permits analysis at higher taxonomic ranks simultaneously, due to the strongly conserved ITS2 secondary structure consisting of its 4 characteristical helices. Thus, the results from secondary structure analysis can improve the phylogenetic resolution obtained from the primary sequence.
Welcome, dear colleagues. My name is Matthias. I'm working in the ITS2 group for about 8 years now. I'm working here at the Department of Bioinformatics at the Biocenter at the University of Würzburg in Germany. Today we would like to present you a little movie on how to use the ITS2 database.
Hi, I'm Jörg Schultz. The reason for that movie is that the ITS2 database is in fact not only a database for storage, but we also provide a lot of tools which allow you to improve your phylogenetic analysis. In this movie, we want to show you how to work with these tools and how to connect all these tools together.
Correct delineation